Hello everyone, this is Arjun and this is my proteomic presentation. So the purpose of this presentation is to explain a protein which I have researched and explained and to discuss the findings within the context of bioprospecting. Before I go into telling the story of the protein, I must reflect on the concept of bioprospecting. So what is bioprospecting? Bioprospecting is the search and exploration of genetic material from plants, animals, and microorganisms from different habitats and biomes, which can be used in practical application in pharmacotherapy, clinical medicine, technology, and in the environment. The aim of bioprospecting is to discover new biological compounds from genes and other useful products from other organisms which can be used to sustain and maintain a better life for all. However, it must be noted that bioprospecting has tremendous responsibility to the environment, to the heritage, and to the cultural protection of the knowledge from which this organism has been researched from. While bioprospecting has the potential to provide valuable new products and technologies, it also is a great controversial issue globally due to the concern over biopiracy and unregulated products for consumer use. As a result, the unauthorized commercialization of traditional knowledge and resources of indigenous people and local communities are not encouraged. And as a result, many countries around the world has put into place strict restriction of how bioprospecting is done. So the question is bioprospecting necessary? And what I found out is yes. The answer is yes. It is a practical reason for doing or to conducting bioprospecting. And here are some successes of bioprospecting which I found during this research. And I have them in four main categories. One, some of the medical breakthroughs for bioprospecting has led to the discovery of many drugs. For example, the drug for anti-cancer, Taxol, and for anti-malaria drug. Agricultural advancement. Bioprospecting has helped in developing of new crop varieties and plant-based products that had led to increased yield and to improve the food security globally. For example, in this instance, there was a new enzymes that was discovered that produce crops that are disease resistant. Thirdly, industrial application. Bioprospecting has led to the discovery of new enzymes and other biological molecules that have numerous industrial applications. A classical example is the discovery of an enzyme from a bacterium found in some hot springs in Japan had led to the development of discovering or putting new technology in how we do our laundry. Fourth, the environmental benefits of bioprospecting has led to the discovery of new organisms and compounds that can be used to clean up pollution and protect the environment. Again, a classical example, they discovered gene from bacteria that can break down oil spills and fungi that can break down plastics through bioprospecting. Therefore, overall, we can see that bioprospecting has the potential to bring numerous benefits to society, including advances in medicine, agriculture, industry, and environmental protection. Now, let's go and look at the protein which I have researched. So this is the story of the protein. So the name of the protein which I found is hemoglobin beta B chain B. It is found in the protein database with the name code 1IWHB and is found in locus 11. It consists of 146 amino acid residues, and its classification is oxygen storage and transport. The sequence or the multilinear attribute method is linear, and below here I have the 146 sequences of this protein. So this protein was discovered in the species called Equus caballus. What is Equus caballus? Equus caballus is the domesticated common horse, right? So chain B hemoglobin is found in this organism for thousands of years. The toxic number for this organism, which is found in the protein bank, is 9796 with a gene symbol HBB. The tissue which this protein has been extracted is in blood, and the method of extraction that has been used is that of X-ray diffraction and serial crystallography. Again, the function of the protein is oxygen storage and transport in the system. Now, let's look at how this protein has enabled horses to survive in their environment. The beta chain protein in horses, or the species Equus caballus, plays a crucial role in enabling the horses to survive in its environment by one. The chain B beta hemoglobin, also which is called in many instances beta hemoglobin, is a protein that plays a crucial role in oxygen transport in the blood of horses. And as we know and discuss in this class, hemoglobin is a complex molecule made up of 4 protein subunits, each of which contain a heme group and binds to oxygen. One of the unique features of chain B hemoglobin in horses is that it has a high affinity for oxygen, which allows horses to extract oxygen efficiently from the air during the air they breathe during respiration. This is particularly important for horses, which are large, powerful animals that require a lot of oxygen due to the power of their muscles. Added to that, chain B hemoglobin in horses has also undergone adaptive evolution, which has led to variation in the protein structure and shape, conformation, and so on, to meet the specific environmental condition in which the organism lives. For example, horses live in high altitude and must have high hemoglobin variant that allow them to better cope with low oxygen level in the atmosphere. Therefore, overall, the unique properties of chain B hemoglobin in horses allow them to efficiently transport oxygen to their tissues, which is essential for their survival in their environment. In addition, it can be added that there is another protein similar to hemoglobin beta B, but it has some unique distinction. One unique adaptation of such protein is the HBC. And how is HBC different from the regular beta chain? This occurs during a substitution of for glutamic acid for lysine in position 26. This substitution reduces the oxygen affinity of hemoglobin. And as a result, this helps the horse in hot, humid environment to conserve oxygen and not to go in a condition in which they have oxygen deprivation. Now look a little bit more into chain beta B hemoglobin. The chain beta chain of hemoglobin is an essential component of oxygen carrying protein found in the red blood cells. In horses, the beta chain of hemoglobin has undergone several genetic adaptation, allowing them to thrive in their environment for thousands of years. And again, in addition to the substitution of the glutamic acid, here is another substitution that allowed their adaptation. And this substitution is where we have histidine has been substituted for aspartic acid in position 7 of the beta chain, which allows for an increase in affinity for oxygen. A little bit more on the survival of the organism. Horses, as we know, are natural athletes and have evolved with an efficient respiratory system, which allows them to take in large volumes of oxygen at different altitudes. As a result, the hemoglobin beta B protein plays a crucial role in this process by ensuring that the oxygen is bound tightly and efficiently to transport oxygen throughout the body. A little bit about the history of this organism, and I want to touch on this bit. The Equus caballus, or the common horse, are commonly known to us as the horse, is a domesticated animal that has been used by humans for thousands of years for transportation, agriculture, and sport. The discovery of the protein Equus caballus is closely linked to the history of hemoglobin research. The first observation of hemoglobin and its role in oxygen were discovered during the 19th century by physiologist Claude Bernard and biochemist Friedrich Messier. In addition to that discovery, there was a German physiologist, German von Helmholtz, proposed that oxygen was carried by a molecule in the blood. And follow up from that assumption, French physiologist Claude Bernard demonstrated that the red color of the blood was due to a protein called hemoglobin. In the early 20th century, scientists began to study the chemical structure of this protein in more details, in where we have Max Brutz and John Kendrew at the Medical Research Council in Cambridge, UK, use X-ray crystallography to determine the 3 dimensions structure of hemoglobin. Both of them earned a Nobel Prize in 1962 for this project. The Equus caballus hemoglobin protein was first sequenced in 1970 using the technique today which we call protein sequencing. This process allows researchers to determine the exact amino acid of proteins, which is composed of 4 subunits, 2 alpha and 2 beta chains. The beta chains of Equus caballus hemoglobin was found to have a unique sequence that differs from other mammalian hemoglobin, suggested that it has evolved to meet the specific needs of this organism. A little bit on the cultural significance of this protein. The discovery of this protein of horses was not pioneered by any specific culture. Rather, it was the result of a collective effort by scientists and researchers from around the world. The study of hemoglobin and its role in oxygen transport began in the 19th century and contributes from scientists such as Hermann von Helmholtz in Germany, Claude Bernard in France, later discovering the 3-dimensional structure of hemoglobin by Max Brutz and John Kendrew. As I said before, the discovery of this protein was a global effort. So there are tremendous effort in terms of scientific collaboration and discoveries. The sequencing of the horse protein started in the 1970s, and as a result of collaborative effort by researchers from different countries, sequencing was carried out by scientists at the University of Wisconsin in the US, while subsequent studies were conducted by researchers in Japan, Sweden, and other countries. Overall, the discovery and the study of the Equus caballus hemoglobin protein has been a global scientific endeavor which contributes from researchers from all around the world. Now, I want to go here into the biochemistry of beta chain protein. Now, the structure. The structure is a tetramer, and we know what the definition of a tetramer is. So, HBB hemoglobin beta chain or horse protein is similar to HBB hemoglobin beta chain found in other mammals, including humans, by having 4 subunits of the heme group. The HBB gene in horses is located on chromosome 11 and encodes the beta globin subunit of hemoglobin, which is a critical component of oxygen in the body. This is the chimera structure, which is ribbon-like. And here we have a little bit of the chimera structure, which I'm just going to highlight to look at some of the structure here. So what we have here. So looking at the 3D view of this protein, while it's loading, what we see here, we can see the structure, we can see the side chains, we can see the amino acids, we can see the peptide bonds and the hydrogen bonds around here. Now, what we also could see here is the hydrophilic and hydrophobic properties of this protein. Now, we can also see the membrane-bound proteins, and those are that within the cytosol of the structure. Right? And as we go around the ribbon here, we see the tetramer. There's 1 monomer, 2 monomer, 3 monomer, 4 monomer. 4 units contribute to the tetramer, and they're all different color scaled. As we move along the ribbon, we can see the different protein and how they're structured. We can also see the heme group. We could also see within each residue whether or not they're polar, non-polar, and the charges on them, and their properties, whether they're membrane bound or whether they're inside the structure. We can see some of the binding sites, so on and so forth. Let's look at another structure. Looking at this structure here, I just want to go into it a little bit more. Again, we see there's a color change here. This protein also shows those side chain that is hydrophobic, and you see it's on the outside. We see the different amino acid, leucine, arginine, glycine, whether they're polar, non-polar, or so on. So there's a lot can be tell from this project about the structure and the function of the protein relates to whether or not they're hydrophobic, hydrophilic, and the charges, and how they conform for their structure and function of oxygen binding or oxygen affinity. So let's go back. All right, so that's my chimera project. Want to go again a little bit, and here we are. I'm gonna make an attempt to go and explain the structure of this protein. So as we know, it has a primary, secondary, tertiary, and quadrant structure. The primary structure of this protein structure is the hemoglobin. The primary structure of this of the hemoglobin beta chain in this organism, it consists of a linear sequence of 146 amino acid, which was mentioned previously, and this is encoded by the beta globin gene. The secondary structure of the hemoglobin beta chain in Equus caballus, or in horses, is composed of 8 alpha helices and 3 beta strands, which are arranged in a specific pattern. The helices and strands are stabilized by hydrogen bonds between the backbone of the atoms to each adjacent amino acid. The primary structure also have the hemoglobin is composed of our 146 amino acid, which was previously mentioned, and is located in chromosome 11. The beta chain contains 8 alpha helices segmented connected by short non-helical segments, and has a heme group bound to each 4 subunits that makes up the hemoglobin molecule. The primary structure of the hemoglobin is critical to its function, and any mutation in the amino acid sequence of this protein can affect the stability and the function of hemoglobin molecule, leading to diseases such as sickle cell anemia and thalassemia. Again, a little bit more of the primary structure. The beta chain of the hemoglobin contains amino acids that are both hydrophilic and hydrophobic, as well as charged amino acids. Some of the hydrophobic amino acid in the beta chain include that of valine, leucine, isoleucine, phenylalanine, and methionine. These amino acids tend to be found in the interior of the protein, away from water molecules. Some of the hydrophilic amino acid in the beta chain include serine, threonine, asparagine, glutamine, lysine. These amino acids tend to be found on the surface of the protein, where they can interact with water molecules and other polar molecules. The charges of the amino acid in beta chain include lysine, arginine, histidine, glutamine, and aspirin. These amino acids can form ionic interaction with other charges of amino acid, or with oppositely charged molecules such as ions and other proteins. The specific arrangement of the hydrophobic and hydrophilic and charge of amino acid in beta chain is critical to the stability and the function of the protein. Any changes or mutation in the amino acid again can cause instability, which lead to disease in the organism. The secondary and tertiary structure. The secondary structure of the beta chain of the hemoglobin consists of alpha helical segments that are connected by short non-helical segments. They are a total of 8 alpha helical segments in the beta chain, with a non-helical segment located between them. Overall, the secondary and tertiary structure of the beta chain of the hemoglobin are critical to its function in binding and transporting oxygen throughout the body. Again, any changes or mutation in the structure or in the secondary and tertiary structure can lead to diseases such as anemia and thalassemia. A little bit more on the tertiary structure. The tertiary structure of the hemoglobin beta chain formed by folding of the secondary structure into a compact globular protein. So this is where the folding of the protein occurs. The folding is primarily driven by hydrophobic interaction between the non-polar amino acid. This is very, very important. And in class we discuss misfolding, and in misfolding also can result in conditions and disease forming in the organism. The hemoglobin tetramer has a complex quaternary structure that enables it to bind and release oxygen in response to changes in the concentration of the blood. And lastly, let's look at the quaternary structure. The heme group contains an iron atoms that binds to oxygen molecule. The 4 subunits are arranged in a quaternary structure, with 2 alpha globulin subunit and 2 beta globin subunits forming a tetramer. The beta globin subunit is essential for oxygen carrying capacity of the hemoglobin. Like humans, mutation in the HBB gene can also result in various types of hemoglobin disease in horses, such as equine infection of anemia, which can cause a range of health problems, and since there will be a reduction in the oxygen capacity in the organism. The molecular shape of this protein, the molecular shape and charges on beta hemoglobin play a critical role in the biochemical function. The shape of the protein determines its ability to bind and release oxygen, while charges in the protein help to stabilize the structure and facilitate interaction with other molecules. The molecular shape of the protein of beta chain hemoglobin is critical for binding and releasing of oxygen. In totality, the protein has a unique shape that allows it to bind to the 4 molecules of oxygen. One of each has 4 subunits. The shape of the protein is determined by the folding of the amino acid sequences in 3-dimensional structure. Again, worthwhile to note, any changes in the amino acid sequence, such as those caused by genetic mutation, can alter the folding and ultimately affect the shape of the protein, which results in diseases. A little bit about the charges. The charges on the beta hemoglobin protein also plays a significant role in its function, such as the stability of the protein. In addition, the charges of the protein can also affect the stability to bind oxygen. Oxygen is a polar molecule with a slight negative charge on one end and a slight positive charge on the other. The charged amino acid on the beta hemoglobin protein can interact with charges on the oxygen molecule, helping to stabilize its binding interaction to the heme group of the protein. Therefore, overall, the molecular shape and charges on the beta hemoglobin are critical to the ability of the main function, and that is to transport oxygen throughout the body of the organism. Comparing a chain B hemoglobin to human hemoglobin. This high degree of similarity between beta chain hemoglobin of horses and human means that they have similar structural and functional properties. They both bind oxygen and transport it throughout the body, and their structure are highly similar at the molecular level. However, there are some differences in the amino acid sequences between the beta chain of the hemoglobin of horses and human. These differences may result in a slight variation in the structure and function of protein, which result in diseases, again, mentioned early on, in thalassemia and anemias. Bioengineering. Some of the practical application of beta chain protein. Beta chain hemoglobin is a protein that is a component of hemoglobin, the molecule responsible for carrying oxygen in the red blood cells. Researchers have been studying beta chain globin for many years and have found quite a few practical functions, and here are some of them. Some clinical application of beta chain hemoglobin B include for diagnostic purposes. Beta chain hemoglobin B can be used as a marker to diagnose equine genetic diseases and disorder related to hemoglobin. What we call blood doping detection, as with other types of hemoglobin, beta chain hemoglobin B can be used as a marker to detect whether the organism has any other steroids or any other material in the body. Beta chain hemoglobin is used during equine transfusion. Beta chain hemoglobin can be used in the transfusion of horses with anemia and other blood disorders. Practical application, as I mentioned before, diagnostic tools to determine genetic disorders such as sickle cell anemia and thalassemia. These are some of few therapeutic applications of beta chain hemoglobin. One, to develop oxygen carriers. Beta chain hemoglobin B can be used as a template to develop oxygen carriers for use in treating human patient with anemia or other oxygen carrying deficiencies. Secondly, treatment of ischemia. Ischemia is a condition where there's inadequate blood flow to organs or tissues. And thirdly, this one is very significant, and it's been used. Drug delivery. Beta chain hemoglobin B can be used as a carrier for drugs and other therapeutic agents, allowing for targeted delivery to cells and organs around the body. Application. Beta chain hemoglobin also used in a laboratory as a research tool. Scientists use protein to study the structure and function of hemoglobin, as well to investigate the effects of mutation in proteins. Researchers are exploring the use of hemoglobin-based oxygen carriers HBOCs. Some of new application research. Drug discoveries. Hemoglobin beta chain has been investigated as a target anti-sickling drug to treat sickle cell diseases. Material sciences. Hemoglobin beta chain has been used to create novel material such as biodegradable hydrogel and nanofibers. Biotechnology. Hemoglobin beta chain B can be used as a model protein for studying protein folding and stability. Some of the novel application or possibility of application for this protein in the future include one, the beta chain horse hemoglobin to create and modify obligate aerobic bacteria to increase the rate of the reaction of decomposition process. Therefore, this protein can be used to modify bacteria to produce biofuel. Using the protein to modify the growth of microorganisms, enhance the process of microbial modification in bioreactors. This process is known as microbial fuel cells, could potentially be used to generate electricity from wastewater and other organic materials. Again, some of the novel application in the summary. In this biomodification engineering, microorganisms such as bacteria are used as a catalyst to break down organic material to generate electrons, which are harnessed and produce electricity. These are the references that I used during this research. Thank you for listening, and good luck to everyone.